Hello and welcome to your Chapter 3 video lecture series on bioenergetics. Here is an outline of all of the topics that we will discuss, starting with cell structure, biological and energy transformation, fuels for exercise, high energy phosphates, bioenergetics, aerobic ATP tally, efficiency of oxidative phosphorylation, control of bioenergetics, and the interaction between aerobic and anaerobic ATP production. So we'll start off introducing what metabolism is. Really, it's all of the chemical reactions that take place in our cell, and within those reactions, we are always going to start with reactants, and those may be combined or broken apart to get a product. So this is just one example of that. And we'll also discuss how one reaction may lead to another, where the product of one reaction can then lead to another. So two types of reactions that we are going to put under the umbrella of metabolism is anabolic reactions, which would be synthesis of molecules. And you could see that illustrated over here where we have smaller molecules or elements here that are going to combine with ATP in order to create a larger molecule. And we can think of the A in there as meaning add. So we're taking these smaller molecules and adding them together to get a larger one. And the second reaction is a catabolic reaction, where we are going to take that C and think of cut. We are going to take the larger molecule, break it down or cut it apart in order to get the smaller components of it. And in that process, we release energy, known as ATP. Now, this entire chapter is about bioenergetics. So this is our process of taking the food that we eat that contains fats, proteins, and carbohydrates and turn it into usable energy so our cell can utilize it for its cell work. So let's talk about the cell a little bit. Our cells are the basic functional units within our body, and they're going to consist of four main elements. We've got oxygen, which makes up about 65%, carbon, that makes up 18%, and hydrogen, which makes up 10%, and nitrogen, which is 3%, all in all making about 95 to 96% of our body. And as far as the structures that make up the cell, there are a ton of organelles, but we're just going to highlight a few of them in reference to the muscle cell. I put a picture of the typical cell that you may be used to studying in biology, but we know that cells come in all shapes and sizes. Think of what a neuron looks like, and of course what a muscle cell looks like. It's elongated, it's not circular like this one here. So when we talk about a cell membrane, we're referring to the boundary of the cell. So here in the muscle cell, it is going to be on the periphery, and we call it the sarcolemma. Sarco means muscle, so anytime we see that, it's in reference to a muscle cell or muscles within the body. And this membrane is going to be semi-permeable, meaning we can't move all items in and out through it. It's going to be particular. Maybe it's going to let water through, but not larger molecules like glucose. So it really helps to create this segregation in the materials outside of the cell versus inside of the cell. The other structure we have is the nucleus, and the nucleus contains genes that regulate protein synthesis, and that will help determine the cell composition and control cellular activity. It is the branch of molecular biology that's concerned with how these genes are being regulated, and we'll dive in deeper in the coming slides on that. And what is unique about our skeletal muscle cell is that it doesn't have just one nucleus like most of our cells. In fact, it has multiple nuclei. So you can see those distributed here on the periphery of this skeletal muscle cell. And that fluid sitting between the nucleus and the cell membrane is called the cytoplasm. This within the muscle cell is called the sarcoplasm. There is that prefix sarco once again, so we know it's referring to our muscle cell. And within this fluid, we're going to see the organelles are suspended within it. And one of those organelles are the mitochondria within here. Now, the mitochondria are the powerhouses of our cell. They work through oxidative conversion of the food into usable cellular energy, And in order to do this, we have to use enzymes. So we'll talk more about enzymes in the coming slides. Notice how in the cytoplasm, or in this case, the sarcoplasm, also has a ton of these contractile proteins, and they are occupying a large portion of the cytoplasm. Now, one of the research pieces discussed in this chapter are the different types of mitochondria. You may have just learned about the mitochondria in general, but within our muscle cell, there are two different types. We have the subsarcolemmal mitochondria. What did sarcolemma mean again? If you said it's in reference to the plasma membrane of the muscle cell, you are correct. And so these mitochondria are located right below the sarcolemma, the cell membrane of the muscle cell. And their job is to produce cellular energy that we need to actively transport ions. And then we have our intermyofibril mitochondria. When we say myofibril, we're really referencing the contractile proteins. So these mitochondria are found near those proteins that we saw filling up that sarcoplasm. And their job is to provide energy for muscle contraction. So when we engage in endurance exercise, we are going to promote a significant increase in mitochondrial biogenesis. And these new mitochondria are constantly going to be produced, especially when we are putting on that stress of endurance exercise. Now, I mentioned we would revisit molecular biology, and this is going to overlap with other areas of biology. Within this field, we are going to study the molecular structures and events underlying biological processes, and we've got relationships between genes and cellular characteristics that they control. The genetic code for specific cellular proteins are brought about through protein synthesis. And so we have an ability to regulate protein synthesis by turning genes off and on. When we go through our exercise training, we can modify this process. For instance, strength training is going to increase the synthesis of our muscle contractile proteins, so we are able to more forcefully contract, producing strength. Molecular biology also provides the tools for understanding the cellular response to exercise. So let's just take a moment to consider these other fields of study such as genetics, exercise physiology, and biochemistry that all play a role in molecular biology. Now let's move into cellular chemical reactions. We have two ways of classifying these reactions. We'll start with our endergonic reactions that are going to require energy to be added to the reactants. So if we look at this graph down here, we can see that our free energy is very low for our reactants, and we are going to have to increase and add energy to these reactants so that the reaction can move forward. So this is not a spontaneous type of reaction. Again, energy is added in order to reach the other side of the reaction and have our products develop. With exergonic reactions, we have the opposite take place. Our reactants are going to release energy, and we could do this in one of two ways. We can do it through combustion, where the energy is released in one single reaction, or through cellular oxidation, where we have a series of steps or smaller reactions that release energy as we go to achieve our end-all products. And you could see that taking place over here in the graph as well. Now, usually we are going to have these reactions kind of hold hands. They are called coupled reactions, where when we have a liberation of energy in an exergonic reaction, that helps drive the endergonic reaction. And that is also illustrated down here by showing these gears. Reactants moving into products. You can imagine this as being the exergonic reaction, and that is helping to drive the endergonic reaction. Excuse me, endergonic reaction from the reactants to the products. Now, continuing our chat on coupled reactions, oxidation reduction reactions are one of the more important types of coupled reactions. Some molecules can act as both reducing and oxidizing agents, but when we want to separate these out, we can look at one as being oxidation. So this is going to be our molecule that is having an electron removed. So the reason here we have this O, meaning it's neutral, and then here we change it to have a plus, is because electrons are negatively charged. So when we remove that electron to later give it to product B here, we are taking that negative charge away from it, and therefore we're left with a positive charge on that atom. And when we say something is going to go through reduction, it means we are adding an electron to it. So here we see molecule B is neutral, but then it gains that electron from molecule A. We can kind of see that in the middle here. And therefore we have one more negatively charged electron, so we're going to put a minus here. So these oxidation and reduction reactions are always coupled reactions. A molecule is losing an electron and we say it is oxidized, and a molecule that gains the electron is reduced. So when I learned this in chemistry, there was actually a little saying, and it was oil, if I'm remembering this right, it was oil rig, and that stood for oxidation is losing an electron and reduction is gaining an electron. So hopefully that kind of helps. And just a quick little side note, you may be looking at the word oxidation here and thinking, oh, it kind of sounds like oxygen, maybe it has something to do with that. And it doesn't mean that oxygen is participating, but it references to oxygen being a electron acceptor. And once we get into some of the different pathways of aerobic respiration, I'll explain why oxygen likes to accept electrons. But the oxidation reduction reactions often will involve a transfer of hydrogen atoms with their electrons. So that's kind of what's happening over here. Next, let's talk about the roles of NAD and FAD in electron transport. So first we have our nicotinamide adenine dinucleotide. That's what NAD stands for. And this is actually derived from the vitamin niacin, which is vitamin B3. And its oxidase form is NAD with the plus here. And you can see that molecule on the right. And this is the site where we're going to have a reaction take place in adding two hydrogens for it to become NADH. And this is now our reduced form. So we've got a hydrogen that attaches here and then a free-floating hydrogen. But essentially these two red dots here are letting us know that two electrons have been added, whereas here we have the plus, meaning that we have one less electron there in that structure. So the other molecule that we have is flavin-adenine dinucleotide, or FAD. This is going to be derived from riboflavin, which is vitamin B2. And oxidized form is FAD, which you could see pictured over here, and the reaction site is right in the middle. Here we are going to add two hydrogens. So we see one will attach to that nitrogen atom, and then we can also have one attached to this nitrogen atom. I forgot to mention that in the previous picture. And this creates FADH2, and that is the reduced form. Both NAD and FAD are going to play an important role in transfer of electrons and are known as carrier molecules when we talk through the bioenergetic reaction. So again, we'll talk a bit more about that when we get into glycolysis, the citric acid cycle, and electron transport chain. Now let's start our next topic on enzymes. Enzymes are going to be these large cellular proteins that are going to act as catalysts to increase the speed of reactions. So they don't necessarily cause the reaction to take place, but again, they can increase the rate and the speed in which that reaction takes place. And they are not going to be consumed within that reaction either. So we are just, by using enzymes, are just increasing that probability of the reaction taking place. So what it does is when we have a reaction that is going to take place without the enzyme being present, there's almost this wall, this amount of activation energy, which is the energy that we need in order to initiate that chemical reaction, that is going to be larger. So that reactant eventually gets enough energy to have that reaction come through and we get our products. When an enzyme is present though, it lowers that activation energy. So it's kind of decreasing the height of that wall, and the reactant can then climb over that wall and reach the other side to create its products. Now, there are certain factors that will regulate enzyme activity, such as temperature and pH, and we'll talk about that in a coming slide. When we have an enzyme present, we need to have the substrates fit almost perfectly, not exactly, but very close to the ridges and crevices within that enzyme. And we'll take a look at this on the next slide here. Here's our enzyme, and you could see there's the ridges and crevices we were talking about and grooves. This is where the active site is. So we have our substrate molecules, and like I said, we want that to fit in pretty well into this enzyme. And then that enzyme along with the substrates creates an enzyme substrate complex. And after that reaction is complete, the enzyme is then going to dissociate, releasing the product. Now this reaction that we saw here with the enzyme is a synthesis type of reaction. Think of it as like an anabolic reaction where we started with smaller molecules and got a larger one. And another type of reaction that is called a degradation type of reaction can take a larger substrate or molecule, place it in the active site, and then break it apart. So we would get these smaller products. And this would be like a catabolic reaction taking place. But again, once that reaction goes through, we are going to have that enzyme dissociate. And notice how these products are not, or I shouldn't say products, these substrates are fitting pretty well in here, right? But sometimes it's not an exact fit, and what the enzyme can do is change its shape so that it can be an induced fit model with those two components interacting with one another in order for the reaction to go through. And then that enzyme is free to be reused again. Now, how do we classify enzymes? All enzymes, well, I shouldn't say all enzyme names, but almost all enzyme names are going to end with ACE. So we've got some examples here, the kinases, dehydrogenases, and so forth, but there are some exceptions in naming the older enzymes that we've left as is. For example, pepsin in the stomach or trypsin in the small intestines. And so let's look at some of these examples of enzymes. First, we have our kinases. And this is when we are going to add a phosphate group. So you can see here we have a substrate and we're combining it with ATP. We know ATP is going to be able to break off its phosphate bonds. And so it does. It gives one phosphate to the substrate while itself turns into ADP. The dehydrogenases are going to remove hydrogen atoms. So we start off with glycerol, and then as our carrier molecule here, the NAD+ comes in, it is going to take off that hydrogen, and we are left with a double bonded oxygen. The oxidases are going to catalyze oxidation reduction reactions involving oxygen. So here, if we look at this molecule, we apply our enzyme glucose oxidase, and that is going to be able to have its oxidation and reduction reactions take place so that we can change the structure of the molecule. Lastly, we have our isomerases, and this is when we're going to have all of the atoms stay within the molecule, but it will be rearranged. So here we have malleate isomerase, and we are going to rearrange those atoms so that we get a completely different structure. Now, what's interesting with enzymes and really valuable is utilizing them to check the levels within the blood to see if there's some type of disease process taking place. So in healthy people, cellular enzymes are not really found within the blood. But when we have certain diseases, and you could see some of them listed over here, we are going to have increases within the enzyme levels in the blood that can serve as biomarkers, showing us that there is disease or tissue damage or dead cells that are taking place within our body. So here's an example. If we have elevated lactate dehydrogenase or creatine kinase within the blood, it may indicate a myocardial infarction or a heart attack. And you could see that taking place over here. We're looking at higher troponin levels and creatine kinase and myoglobin as well. So when we are having a heart attack, troponin actually is going to increase within 2 to 3 hours, whereas creatine phosphokinase or CPK is going to rise within 6 to 12 hours. These may also be biomarkers for things like acute coronary syndrome or ischemia as well. I mentioned that we would talk about the factors that influence enzyme activity, such as temperature and pH, and here we are. So when we have a small rise in the body temperature, we are going to increase enzyme activity. And if you think about when we're exercising, that helps to raise our body temperature. So when we have larger increases, though, above 41 degrees Celsius, that can denature our enzymes and decrease the activity. So it is a fine little window that we want to stay within of when our enzymes can function optimally. This is also why when we are sick, our body temperature rises. One of the reasons is our enzymes can be more productive at higher temperatures. But of course, you don't want that fever to get too high. And then we have pH. So when we have an increase or decrease, that can decrease enzyme activity. So when we do high intensity exercises, that is going to decrease our muscle pH. We will get a buildup of hydrogen atoms. And then you could see in this graph that if we go above, let's say 7.5, we are going to start to see a decrease in that enzyme activity. And of course, headed over closer to 9, that's incompatible with life, right? They're going to denature. Now let's get into the fuels for exercise, starting with carbohydrates. Our carbohydrates are made up of carbon and hydrogen and oxygen atoms. We do have 3 different forms. We have our monosaccharides, which are just going to be our simple sugars. So mono means 1. We'll have 1 ring for these sugars. Glucose would be our blood sugar, and fructose could be found in fruits or honey. The other type we have is a disaccharide. This means we have 2 sugar molecules attached to one another. In this case of sucrose, we have glucose and fructose coming together to create table sugar. We can also find this in cane sugar, beets, honey, and maple syrup. And the last form is going to be a polysaccharide. And this is best demonstrated with glycogen down over here. That is going to have hundreds to thousands of glucose molecules bonded to one another. And this molecule is created so that we can store it for fuel in the liver and within our skeletal muscles. And it's synthesized by the enzyme glycogen synthase. Now you might be thinking, why the liver and muscle cells? Why don't we just store individual glucose molecules within our cells? And the reason for that is because of the osmotic pressure that would take place. If we put all of the individual glucose within our cells, water is going to want to travel to that area and our cells would swell and eventually lyse or burst. So that is why we rather store it as a larger molecule within the liver or our muscle cells. Although these stores are small, they can quickly be depleted within a few hours from prolonged exercise. And we are going to use the process of glycogenolysis to break down glycogen into the individual glucose molecules. Now, those of you that do low carb diets, you might hamper the glycogen synthesis process. Whereas if you have a high carb diet or a regular American diet, really, that will enhance glycogen synthesis because we'll have all of this glucose within our blood that we want to store. Next, let's talk about fats as being a fuel for exercise. Fats are going to be insoluble in water, and we do have a greater ratio of carbon to oxygen than we do with our carbohydrates. And there are 4 different groups of fats. We have our fatty acids, triglycerides, phospholipids, and steroids. Now, the fatty acids are the primary type of fat that is used by skeletal muscle. They are just these chains of carbon atoms linked to carboxyl groups at one end, and you could see that they have different structures, whether they're saturated, monounsaturated, or polyunsaturated. The triglycerides are the storage form of fat, really within the fatty acid tails here, within the muscle cells and adipose tissue. And then they have glycerol attached to it, which is an alcohol, and that could be used by our liver to make glucose. And this is, like we said, the best storage form within our fat cells. So when we exercise and we need to turn to fat to create energy, we can use the process of lipolysis that is regulated by the enzyme lipase to break that down and utilize it for energy. Then we've got our phospholipids. Now, these are not really used as an energy source, but we do find them within every cell. We're going to have our phospholipids have its phosphate head, and then we've got the fatty acid tails here. They're going to help create that plasma membrane and keep structural integrity, and it also is going to provide insulating sheaths around our nerve fibers. Steroids are derived from cholesterol, so this is not an energy source, but we are going to use it within our cell membrane to create a little bit more fluidity within it. And it's also going to be needed to synthesize sex hormones such as estrogen, progesterone, and testosterone. And you could see what that looks like. We have these 4 rings put together. Now let's get to protein. Proteins are composed of amino acids. So when we have our full protein, you could see it's folded up like this, and then we can pull some of those bonds apart and we get a polypeptide chain. If we further break those polypeptide chains apart, breaking their peptide bonds, we will eventually end up with amino acids. So here is a chart of all of the different amino acids that there are. There's 20 in total, and these could be used as fuel in several ways. We have one specific amino acid, alanine, that can be converted to glucose within the liver and then eventually stored as glycogen. So this process is called gluconeogenesis. Then other amino acids can be converted to metabolic intermediates, and these would include isoleucine, alanine, leucine, and valine. And these intermediates can then be used within the citric acid cycle and can contribute as fuel in the muscle. But this is rarely used as a big piece of fuel for our muscles during exercise. They will really only contribute to 3 to 15% max of our fuel for our muscles. And 1 gram of protein is going to give us about 4 kcals of energy. I forgot to mention this in the previous slide, so let me go back. 1 gram of fat will make up 9 kcals of energy, and for our carbs, it is 4 as well, just like protein. So let's now move on into our high energy phosphates. Our energy currency within our body is adenosine triphosphate, or ATP, and we can see the full molecule over here. It is consisting of an adenine molecule, a ribose sugar, and then we've got 3 linked phosphates. And of course, as we break off those phosphates, we're going to release energy. So if I break off one, this is now called ADP, or adenosine diphosphate. And if I were to further break off another one, then this would be called AMP, or adenosine monophosphate, mono meaning one. An ATP is an immediate source for muscle contraction, and our cells without significant amounts of ATP are going to die quickly. So how do we get that energy out? We are going to use the enzyme known as ATPase to break these high energy bonds in order to release it. And here is a figure depicting that cells use an exergonic reaction to break down our food in order to form ATP via endergonic reactions. So we will produce that ATP and then use that for cellular work. The ATP is driving the energy requiring cell work. Now we can get into the topic of bioenergetics. We have our formation of ATP that is going to take place through metabolic pathways, and these pathways exist in our cells to produce ATP rapidly because all of our muscles need them continuously. So those three metabolic pathways are phosphocreatine breakdown, our degradation of glucose and glycogen, which is also known as glycolysis. And these first two pathways do not involve oxygen, so they are called anaerobic pathways. And then we've got our oxidative formation of ATP. This is going to utilize oxygen, so we call it an aerobic pathway. And that's essentially what's being said down over here. So we'll start off with our anaerobic pathways. With the ATP-PC system, this is also known as our phosphagen system. This system gives us a really rapid way of making ATP by having a donation of a phosphate group. Our muscle is only storing a small amount of PC. PC stands for phosphocreatine, which you can see the spelling over here. And we'll take the phosphate from this molecule and add it to ADP using the enzyme creatine kinase. And in this way, we get ATP, and what is left over is creatine. So this reaction is limited because we've only got so much phosphocreatine. It'll provide energy at the onset of our exercise, so it's short term and for high intensity exercises, and really will help us during the first 5 to 15 seconds of our exercise. So examples of exercises that would utilize this system are sprinting 100 meters, high jumping, rapid weightlifting, American football player racing 10 yards downfield, really short, intense bursts of exercise. And in order to come back and recreate these phosphocreatines, we are going to require ATP to be present, and this will reset during the recovery from exercise. So there was a little segment here on the winning edge when we talk about creatine supplementation, and this can help improve exercise performance. Studies showed that having about 20 grams a day of creatine monohydrate over 5 days increased stores in muscle. There was also improved performance in lab settings for short duration, meaning it was about 30 to 180 seconds, and the high intensity exercise. When the subjects performed resistance exercise, they also saw that it resulted in enhanced physiological adaptation to weight training, and it promoted increase in dynamic muscular strength as well as fat-free mass. So while this is all in good, there are some possible side effects that include nausea, neurological dysfunction, minor GI upset, and muscle cramping. But these side effects are not well documented. So there have been many studies that have been done, both that have been short-term and long-term studies, as much as 5 years, which have indicated that they are safe and well tolerated for people to use. But there's also an issue within the different supplements that are offered, in some being contaminated and containing other chemical compounds, being pulling in substances such as filler within the supplements. So there is a high variability in the purity of the supplementation. Now let's go back to some more anaerobic ATP production, and this time we're going to talk about glycolysis. This is when we take a glucose molecule, or we can also utilize glycogen from the muscle or the liver, and we are going to be able to create 2 pyruvic acid or pyruvate or 2 lactic acid or lactate molecules. Now, in order to go through this process, we do have to invest 2 ATP molecules. It's kind of that thought process of it takes money to make money, right? You got to put in some energy to get new and more energy out. So all of these reactions are going to be enzymatically catalyzed through coupled reactions, and it takes place in the sarcoplasm of the muscle. We break it down into 2 phases. We've got the energy investment phase, where you're seeing the 2 ATP being utilized, and then we have our energy generation phase, where we are going to be able to produce 4 ATP, 2 NADH, and 2 pyruvate or lactate molecules. So all in all, this is going to give us a net gain of 2 molecules of ATP. Now we'll get into what pyruvate is in a little bit, but you may have already heard of the molecules lactic acid or lactate, and sometimes they're even used interchangeably. So what I wanted to clarify is that lactate is actually the base of lactic acid. So you could see here that hydrogen has been removed. And when acids dissociate, they release their hydrogen ions. That's why we see it here. So we're going to call lactate the conjugate base here. Now let's start getting into the details of glycolysis. So there are 2 starting points. We can either use glucose from our blood, which then we have to utilize ATP in order to phosphorylate it to turn it into glucose 6-phosphate. But if we're starting with glycogen from the liver, it has to be phosphorylated, but we don't have to spend 1 ATP molecule in order to get it to turn into glucose 6-phosphate. Instead, we can use an inorganic phosphate that is in the cell itself. So if we start with glycogen, then through the process of glycolysis, we will actually end up with 3 ATP as a net result rather than 2 ATP, because we are utilizing one of the ATPs here with our blood glucose. So here are all of the steps involved in glycolysis. Yes, this is a lot that is going on here, but we are going to break it down step by step. So in this version, we are going to utilize glucose from our blood glucose and utilize 1 ATP in order to turn it to our glucose 6-phosphate, as we saw in the previous slide. From there, we are going to rearrange our molecules and get fructose 6-phosphate, and then invest another ATP molecule so that we can get to fructose 1,6-bis-phosphate. We are then going to split this 6-carbon molecule here of fructose 1,6-bis-phosphate into 2 separate 3-carbon glyceraldehyde 3-phosphate molecules. And at this point, we are going to start dealing with our carrier molecules. Do you remember what they are? They are NAD and FAD. So here you can see phosphorylation taking place on either side, as well as our carrier molecule coming in and taking those hydrogens. The hydrogens are removed so that later they can be used for generation of ATP in the mitochondria if oxygen is present. Then once we have that process go through, we are going to change this molecule's name to 1,3-bisphosphoglycerate. And here in the red bond, you can see that we are going to remove that and add it to ADP in order to create our first ATP molecule. So that results in 2 ATP molecules here. From removing that phosphate, we are left with 3-phosphoglycerate, and you could see we've got 3 carbons with a phosphate attached. And at this point, we are going to remove water from the molecule, so 2 hydrogen atoms and 1 oxygen in both of these molecules, which will result in our PEP, phosphoenol pyruvate. At that point, we still have that phosphate attached, which you can break off and utilize that phosphate to add to ADP, and that is going to result in 2 more ATP molecules. So at this point, we invested 2 ATP molecules, and we were able to produce a total of 4 ATP molecules. So our net result of going through this entire glycolysis process is 2 ATPs. Now, when oxygen is present, we can shuttle and move some of these carrier molecules to the mitochondria so we can further utilize them to create more ATP. So let's talk about those electron carrier molecules some more. Like we said, they're going to help transport those hydrogens and their associated electrons. Now I threw some more images on here to go back to talking about hydrogen structure. Hydrogen is going to have 1 proton, 1 neutron here, and then we have that electron, 1 electron moving around. Now, if you've taken chemistry before, you know that there's a special rule with how these electrons like to distribute themselves. In this first shell, hydrogen wants to have another electron for it to be happy and stable. So right now, what it really wants to do is find another electron. This is why when there's 2 hydrogen atoms, they are going to share electrons and create a bond. So here's that chemical bond right here, covalent bond, and you could see them sharing, each hydrogen has 1 electron, right? So they're each sharing their electrons and they're stable and happy. So this is why we can attach those hydrogens to NAD and FAD. So to the mitochondria for ATP generation, we send over the carrier molecules. And what we're going to see is after the process of glycolysis, we are able to utilize the pyruvate or pyruvic acid. We may convert this over to lactate as well, or lactic acid, in this anaerobic process, but eventually what we want to do is send over molecules to the mitochondria to go into the citric acid cycle. So what we saw in the process of glycolysis was that NAD coming in, taking 2 hydrogen atoms and utilizing them, right? And we didn't see FAD in glycolysis, but we're going to see it in the citric acid cycle. So this is just a reminder of the chemical reaction that takes place when 2 hydrogens are present. I did have to fix that really quick, that little typo. So when we have it present, it turns into FADH2. So like we said, that NADH from the glycolysis process is going to be shuttled into the mitochondria. And as we send it over there, we want to turn this back into NAD. And we do this by taking pyruvate or pyruvic acid and utilizing an enzyme known as LDH, lactate dehydrogenase, to turn it into lactate or lactic acid. And so what we're really doing is shuttling the hydrogen ions into the mitochondria. And the reason we do this, the reason for this lactate formation, is the recycling of NAD. So that glycolysis is the breakdown of glucose into pyruvate or lactic acid, and we end up, after going through glycolysis, with 2 to 3 ATP. And you could see that process taking place in this illustration too. We have glucose come in. We're in the cytoplasm of the cell right now. We go through glycolysis and create pyruvate, and then we can take that NADH and utilize our enzyme to end up with lactate and our NAD regenerated. That lactic acid can then exit the cell. But if oxygen is present, then we can go back into pyruvate and enter into the mitochondria to have our next step take place, the citric acid cycle. After going through that cycle, we can then enter the electron chain transport that you see illustrated over here. And here's that closer look of an ADH shuttling into the mitochondria. Like we said, it has to be converted back to NAD in order to continue glycolysis, and that pyruvate is accepting the hydrogens and forms lactate. So now let's talk about aerobic ATP production. Aerobic ATP is going to result from the cooperation between citric acid cycle and the electron transport chain. And this is taking place within the mitochondria. This is why we see the cell here. Glucose is taking place in the systole, and here we have the rest of the process taking place in the mitochondria. So there's going to be these two metabolic pathways. Like we said, one of them is the citric acid cycle. This is also called the Krebs cycle or the tricarboxylic acid cycle. So that's why sometimes you see it abbreviated as TAC, but really it's all the same thing. It was called Krebs cycle because it was discovered, and he really pioneered the research here. And that was the scientist Hans Krebs. So what this cycle does is it really completes the oxidation of our fuels using our fats, our carbohydrates, and proteins to provide electrons for our next pathway, which is the electron transport chain. All in all, the main goal of the citric acid cycle is to complete oxidation of acetyl-CoA, meaning we're removing those electrons so we can use them again for the electron transport chain. This pathway is going to have energy obtained from electron transport so that we can utilize it to produce ATP at the end of the chain. So let's dive into more detail on both of these processes. First, we have our citric acid cycle, and this is going to start with our pyruvic acids. Remember, we got that as an end result of glycolysis. And we are going to have each of these molecules enter in separately and convert it to acetyl-CoA. So you can see acetyl-CoA over here. You can see that it involves 2 carbon atoms. And as a result of this conversion, we are going to have 2 carbon dioxide molecules as well. So where did we get this acetyl-CoA from? It can be formed from our carbs, fats, or proteins using NAD and FAD as electron or energy carriers. These electrons that it has contain potential energy that we harvested from the food that we ate. And then the energy will be used in the electron transport chain. And we'll talk about some of the other processes such as fat or protein and how we got to this point. So once we do have our acetyl-CoA though, we are going to combine it with oxaloacetate, which has 4 carbon molecules, and use that to form citric acid. So it makes sense, right? We've got 2 molecules here, acetyl-CoA, 4 carbon molecules here at our oxaloacetic acid, and so 2 plus 4 equals 6. So now we have a 6 carbon molecule, and we are going to then take off 2 carbon dioxide molecules off there. So we had 6 carbons, right? So we're going to pluck off 2, and then we are going to end up with citrate. And citric acid, by the way, is where we got the name for the cycle, if you didn't guess before. And so by utilizing that, we are able to generate 1 ATP and release more high energy electrons. So you can see all of the high energy electrons from each of these processes are all being funneled to the electron transport chain, which we're going to talk about later. So here is another look at this citric acid cycle. Just as a closer view, you can see the specific enzyme names. We're not going to go through all of that and the specific molecule names, but we are going to highlight just a few processes in here. So each glucose molecule is going to result in 2 turns through the citric acid cycle in order to remove electrons and produce 2 ATP. We will have 2 carbon dioxide molecules, and we will end up with 8 hydrogen molecules with high energy electrons. So within those turns, you can see from this illustration that we are going to have NADH as a result, and each NADH molecule is going to later yield 2.5 ATP, whereas our FADH is going to yield 1.5 ATP. What this means is FADH is not as energy rich as NADH. You'll also see as we're going through this cycle that we have one GTP, which stands for guanosine triphosphate, result. And by breaking off one of its phosphates, this is a triphosphate, right? So this molecule has 3 phosphates on it. By plugging off one of those phosphates and giving it to ADP, we can produce ATP. So there's another molecule of energy gained. Now, before we move into the electron transport chain, let's talk about the other food molecules that we can utilize in order to go through this process. So first, we're going to talk about beta oxidation by utilizing fats. Triglycerides can be found in our fat cells or in our muscle cells. Our muscle cells are going to contain enzymes to break the fat down, and that breaks it down into or utilizes our triglycerides to break it down into glycerol, which can be used as fuel, but like we said, it kind of plays a very small role. And we can utilize those fatty acids to convert it into acetyl-CoA. And this process is a beta oxidation. And you could see that illustrated over here, where we end up with acetyl-CoA. So beta oxidation is that process of oxidizing fatty acids in order to form the acetyl-CoA. And it does this through a bunch of steps. We won't go through the details, but essentially the fatty acids are going to get chopped, forming 2 carbon fragments, forming acetyl-CoA, and we can then use it as fuel into the citric acid cycle. Now, what about protein? This image here is depicting for us protein and also the other macromolecules, let's say. So overall, it's an illustration of the interactions between the metabolic breakdown of our fats, our carbs, and our proteins. And this is known as the metabolic pool. We all know that we're not just eating pure protein, right? We get lots of different macromolecules from eating our foods. So like we said before, protein is going to just play a very small role as far as fuel for our exercise. It'll be between 2 or 3 to 15% to fuel our exercise. And that really is going to depend on the amino acids that are present, right? And what molecules we can convert them into. So when we break protein down, the subunits of that are going to be those amino acids that we talked about before. And then it could be converted to glucose. So that's why we have these arrows kind of going back and forth here. It can also be converted to pyruvic acid or acetyl-CoA if we have that alanine, and the Krebs cycle intermediates. So here we can see it transforming into a 6-carbon molecule. Now we are going to get into the second aerobic ATP production process, which is the electron transport chain. This is also known as the respiratory chain or the cytochrome chain. And essentially what's taking place here is oxidative phosphorylation within the mitochondria. So this is the inside of the mitochondria, which we are going to call the matrix. This is the inner membrane space because the mitochondria has a double membrane. And this is going to be our outer membrane, which means this is our extracellular space. Outside of the mitochondria, we're in the systole of the cell here. So now that we have orientated ourselves, the main goal of this process is to use potential energy in our NADH as well as our FADH to rephosphorylate ADP, which you can see taking place over here, and turning it into ATP. So how do we do this? We are going to remove the electrons from NADH and FADH and pass it along a series of carriers, which we call cytochromes. And we pump all of the hydrogen into this intermediate space. Now remember what we said before, our NADH is going to produce 2.5 ATP, FADH produces 1.5 ADP, so NADH is going to be more energy rich. And there's a chemiosmotic hypothesis whereby the hydrogens from the NADH and FADH is then going to be combined with oxygen in order to form water. So we're going to touch more on that in this next slide over here. So we just mentioned in the previous slide that our hydrogen atoms are building up here in the intermembrane space. And this is creating a strong drive because of this increased concentration, the electrochemical gradient that is taking place here. So we have the strong drive for the hydrogen to want to get back here into the matrix. And the intermitochondrial space here, or the membrane rather, is not permeable to hydrogen, meaning it's not going to let hydrogen just move on through. So we need a specialized hydrogen channel called a respiratory assembly to move the hydrogen atoms through it. And this movement is going to activate an enzyme known as ATP synthase, which is going to cause the reaction of ADP to become phosphorylated and result in more ATP. Now, our last cytochrome here is remaining within a reduced state, and we're unable to accept more electrons if oxygen is not present. This whole process stops. But when oxygen is present, the last cytochrome is going to be oxidized by oxygen. We are getting this oxygen from the air that we breathe. And like we said, oxygen has a high capacity to accept electrons. Now, why is that? Let's go back to some chemistry. Like we said, that first shell, it is going to be happiest when we have 2 electrons to fill it. So this first electron is stable, it's satisfied. But oxygen itself normally is going to have 8 electrons. So with its remaining electrons, which are 6, they are going to go in a second ring. And so beyond the first shell, there is something called the octet rule, where these elements are the most happy when they have 8 electrons in their last shell, which is called the valence shell. So we only have 6, and oxygen is craving 2 more electrons in order to be satisfied and stable. And therefore, it is the final electron acceptor. It will take those electrons that the mitochondria has to offer. And as a result, we are going to get water take place. So here is our last cytochrome, and you could see our 2 hydrogens that pumped in through our enzyme ATP synthase, and oxygen being present here combine, and we get water as a result. Now, as we are going through these processes, we are going to start to see an increased production of free radicals. Free radicals, or radicals, are molecules with an unpaired electron within that outer orbital. So like we said, that last shell likes to have 8 of them in there, right? So the free radicals can be produced by the leakage of electrons along that electron transport chain, and it can be produced in other locations within the cell as well. Now, we don't want these free radicals because they could start to react or interact with other molecules in the cell, and that can result in damaging molecules and creating tissue damage or molecular damage, for instance, damaging our lipids, damaging our proteins and things like that. So here's the normal process where those free radicals are kind of kept in check. But when we start to have a buildup of those free radicals, we start to see that we've got increased damage. We'll also have less ATP being generated because we're losing all these electrons, right? It used to be thought that exercise promotes the production of free radicals within working muscles, but what we found is that the free radical production usually is taking place when we are at rest and not while we are exercising. So now that we've gone through all of the aerobic processes, let's do a tally and breakdown from utilizing one molecule of glucose. So within the process of glycolysis, we saw that we were going to have a net gain of 2 ATP. Remember that this is actually going to be 3 ATP if we start off with glycogen, but we will just assume that we're using glucose in this example. We also were able to obtain 2 NADH within glycolysis, and later on, because our conversion of this will be 2.5 ATP, it is eventually going to result in gaining 5 ATP. So if we do continue into that aerobic process of the citric acid cycle and electron transport chain, then we are going to have 7 ATP total at this point. Then we are going to go through the citric acid cycle and take our 2 NADH produced from pyruvate and convert it into acetyl-CoA, and that is going to produce 5 more ATP for us. So our total count is now 12. We also, in that cycle, were able to obtain 2 GTP. Remember, we broke off those phosphates and gave them to ADP, so we can gain 2 more ATP. So now our total is going to be 14. And then we got our 6 NADH and our 2 FADH from that one glucose molecule, which is then going to result in 15 and 3 ATP respectively, and that brings our total to 32 ATP. Now remember, keep in mind, if we did start off with glycogen, we would have 1 extra. So if we start off with glycogen, our grand total would be 33 ATP. So how efficient is oxidative phosphorylation as a system of converting energy from food into usable energy? So 1 mole of ATP, which is really 1 gram molecular weight of ATP, is going to be 7.3 kcals, and 32 moles of ATP, which we got from our previous tally, is going to be formed from 1 mole of glucose. And then our potential energy released from 1 mole of glucose is 686 kcals per mole. So to figure out the efficiency, we are going to take the 32 moles of ATP and multiply it by how much our 1 mole of ATP is, which is 7.3 kcals per mole, and divide that by our potential energy release. And that is going to give an overall efficiency of 34%, meaning that 66% of our energy is being released as heat. So that's pretty efficient. Now, how do we regulate bioenergetics? We are going to want to utilize rate-limiting enzymes, and that's because each of our pathways has a ton of reactions taking place, and we're using specific enzymes, right? And if we just were to let that process continue on, we might have a lot of our substrate, meaning our starting materials, and the reactions would drive forward, and we'd end up with too much product building up, and that would probably not be as efficient. So if we have a rate limiting enzyme, which is an enzyme that's regulating the rate of that metabolic pathway, then we can control it a little bit better. And usually these rate limiting enzymes are going to be at the beginning of a reaction. These enzymes are usually regulated by a modulator, which is going to be a substance that increases or decreases enzymatic activity. And any enzyme that has one of these modulators is called an allosteric enzyme. And we have certain stimulators and inhibitors that will assist us in creating or allowing these pathways to continue. So one of the most common ways is by looking at the levels of ATP as well as ADP and inorganic phosphates. typically high levels of ATP are going to inhibit pathways that lead to ATP production. And when we have low levels of ATP and we start to see high levels of ADP and inorganic phosphate, we need more ATP, right? So that's going to stimulate the pathways for ATP production. One of the more simple examples of this is the ATP-PC system. So remember, this was our phosphagen system where we utilized our phosphocreatine molecule. So I'm going to bring you back to this over here. Our phosphocreatine molecule was added to ADP. We broke off that phosphate and gave it to ATP. So whenever we have a lot of ADP present, this pathway or this reaction is going to take place for us to make more ATP. But if we see that we already have enough ATP, then it's going to inhibit this pathway and we are not going to have to utilize our enzyme to have this reaction move forward. So this is also going to take place in other pathways that you see listed here. And a lot of these are negative feedback loops. We are looking for that homeostasis to kind of go off kilter so that we can have the pathway take place. So our next example is through the pathway of glycolysis using our rate-limiting enzyme of phosphofructokinase. And we didn't go into detail as far as the enzymes that were used in glycolysis, but just know that it's an enzyme that is used in the beginning of glycolysis. So when we start exercising and we start to see that the ADP and inorganic phosphate levels are increasing, that is going to enhance the PFK use in order to allow the pathway to continue forward. Conversely, if we have high levels of ATP, then we're going to decrease the PFK activity. It's going to be inhibited. Another thing that we see is that when we start to see high amounts of hydrogen ions, which is going to decrease our pH, or we start to see that citrate has increased, that is going to inhibit PFK activity as well. And we have some similar examples over here for the citric acid cycle and electron transport chain. And the last thing I'll mention on this slide is that when our muscles contract, when we're exercising, well, we know that calcium is going to be released from the sarcoplasmic reticulum to get our contractile filaments to create its cross bridges, right? And so we know that we have a rise in calcium due to this, and this is going to indirectly activate an enzyme known as phosphorylase, which is going to break down glycogen to glucose so that it can enter glycolysis. So when we have calcium present, that is going to stimulate aerobic ATP production because we want to create more ATP to replace what we have lost. And our last topic for this chapter is to discuss the interaction between aerobic and anaerobic ATP production. So we know that energy is needed to perform most types of exercise with a combination of anaerobic and aerobic sources. ATP production by utilizing the creatine system, glycolysis, oxidative phosphorylation actually takes place simultaneously. I know in this lecture we talked about, first we do creatine phosphate, then our glycolysis, and then the citric acid cycle, and then electron transport chain and so forth. But really this is all happening at the same time as we're contracting our muscles to perform our exercise. But as far as what is being used when, it's going to depend on the duration and the intensity of the exercise. So we've already kind of mentioned when we talked about our phosphagen system or our creatine system is when we do short-term high intensity activities, we are going to have more of our anaerobic energy systems contributing more. So we've got, depending on the duration of that, utilizing them here. And you could see some of those activities on the right-hand side here where it says anaerobic. And then when we have more long-term or low to moderate intensity exercises, you could see the shift where we are going to use a greater percentage of our aerobic energy systems to create ATP. And some of those activities are listed here on the left side. So that is all of our topics for Chapter 3. Please let me know if you want any clarification or have any questions.